Alright, so in this B part, or B chunk of this lecture series, the Lecture 4 series, we're going to continue our discussion of enzyme kinetics. Please remember that in the first part of this lecture series, we talked about enzyme kinetics and thermodynamics in general. We focused a lot of our time talking about making the universe happy with releasing free energy and increasing entropy, making the universe sad with positive delta G's and absorbing free energy from the environment. Now we're going to talk a little bit more about what enzymes do. We left that lecture by making the point that enzymes speed up and make biochemical reactions occur more readily and more rapidly, more easily, by lowering the energy of activation. But we're going to talk about that quite a bit more and put some meat on those bones. And then in the last chunk of this lecture series, we'll talk about how enzymes bind to reactants. We'll then call those reactants substrate, enzyme substrate binding, and how that plays into catalysis. So let's kind of back up a slide or two from the last lecture, just so that we're all familiar. I don't know how much time has passed since you watched that last chunk. So we did talk about at the end of that first chunk A, that enzymes speed up biochemical reactions. They influence the kinetics of reactions by making those reactions easier to occur. By lowering the energy of activation and removing that mountain of energy between products and reactants, they sped up the ability for the reaction to proceed to product. What the enzyme does not change is delta G. It does not change the end product of the reaction. It doesn't change the products themselves. You get there either way, nor does it alter the reactants. It simply changes the energy of activation. It affects the path from products to reactants, or reactants to products. So we looked at this free energy diagram in that first chunk. What we are saying is where you start and where you end is the same whether an enzyme is present or absent. The only thing the enzyme is doing through catalysis is lowering or easing the path between the two, making it easier and more quickly to get from reactants to products. Essentially, it is lowering the amount of energy needed by the universe to invest in order to make this reaction proceed. And we had that analogy of getting from one hotel to another with Mount Everest in the way. So you get the same products either way. The same reaction occurs with or without an enzyme. All the enzyme is doing is lowering the energy of activation. So let's look at this a little bit more. There's a part of this diagram that we haven't yet discussed. It is at that energy peak, and we see that it's referred to as the transition state. We've already said that enzymes don't affect delta G. They don't affect the change in free energy. They don't affect reactants and products, and so your delta G remains the same whether you have an enzyme present or not. All enzymes affect is delta G double dagger, the free energy of activation, the amount of energy required to get to those products. So catalyzed or not, why do we need an energy of activation in the first place? What is delta G double dagger? Why do we need to put energy into a reaction in order to get that reaction to occur? Well, if we think about it, chemical reactions alter reactants and turn them into products. That's really all a chemical reaction is. You start with A, and a chemical reaction changes that into B. If we think about it a little bit more deeply, and we think about it realistically, what's really happening to A, what's really happening to our reactants, is that the reactant molecules are being pulled and stretched and distorted and pushed and ripped at and yanked at and hammered and pummeled and squeezed until eventually product A, or reactant A, becomes product B. We are altering molecules, and that's not a pretty picture. These molecules are being pretty heavily distorted and manipulated so that they can be changed into something else. All of those things, pulled, stretched, pushed, squeezed, hammered, pummeled, and ripped, require energy. That is work to do that. When all that energy of work, when all of that hammering and pulling and pummeling and stretching has been done, that's when we reach the peak of this diagram. So you can think of it as I start with reactants. There's some amount of energy within the covalent bonds of those reactant molecules. There's energy within those molecules. And leaving that energy alone, I'm going to work at this molecule. I'm going to pull on it, which takes work, and hammer it, and squeeze it, and pummel it, and stretch it. All that takes energy. And I'm going to get it to a point where it's barely holding on to itself, and once I get it up here, that molecule is going to crack and collapse into what I want it to become, which is product. When that collapsing occurs, all that energy of distortion comes out again, as does the free energy of the reaction itself as those reactants collapse into product. At the peak here, physically now, the thing that we have created as we've manipulated and stretched and pummeled on reactants is called the transition state. The transition state isn't an energetic concept. That's delta G double dagger. The transition state is a physical thing we have created in the path of this reaction. We make transition state as we convert products into reactants. So we can say that transition state is a hybrid structure that is no longer reactant molecules, not quite yet product, but kind of an in-between molecule that's on its way to becoming product. Remember the bread machine analogy I used for protein structure and enzymes? And I told you it's kind of a weak analogy here, but I'm going to come back to it and it's going to be much more helpful. Well, we're there now. We're back to it. So the bread machine makes bread for us. It makes bread much more easily than we can make it ourselves. We can make bread from scratch, but it takes a lot of work. The bread machine does that work for us, so it's like the enzyme, making it happen more rapidly and more easily. But if the reaction is going from the raw ingredients to bread, then water, flour, salt, and sometimes eggs would be our reactants. Freshly baked bread, of course, is the product. And it's fair to say that the product is nothing like the reactants. I don't know about you, but I don't want to fill my mouth right now with water, flour, salt, and eggs. Sounds pretty disgusting. But I love me some fresh baked bread. So it's fair to say that fresh baked bread is not the same as the raw reactants or ingredients that went into it. If water, flour, salt, and eggs are the products, and freshly baked bread is if water, flour, eggs, and salt are the reactants, and freshly baked bread is the product, then this, right here, is our transition state. It is certainly not water, flour, eggs, and salt. It is not reactants. But there's no way you're going to tell me that this lump of dough is fresh baked bread. It's an in-between state. From one to the other, it's a hybrid physical state that isn't quite product, certainly is not any more reactant, but is on its way to being what it seems to become. That's what our transition state is. Now think about it. You and I could take water, flour, salt, and eggs and make dough, but it would take a huge energy investment to do so. We'd have to knead the dough, mix the ingredients, pull the dough, and stretch the dough, and that takes a lot of work. If we throw everything into a bread machine, the enzyme does the work for us. The enzyme makes transition state much more easily and much more quickly than we can. So with this example in mind, let's define the energy of activation a little bit better. The energy of activation, that is, delta G double dagger, is the amount of free energy needed to make the transition state. Once you make the transition state, the rest of the reaction is easy, literally. Once you've made the bread dough, wait for it to rise and throw it in the oven. You're essentially done. It's making the transition state that's hard. And that gets us to the idea of enzyme kinetics. Enzyme kinetics refers to the speed or rate at which a reaction can occur. We usually determine this rate experimentally, and we express it in terms of the change in concentration of the reactants or the change in concentration of the products per unit time. If we're thinking about kinetics and we're thinking about reaction rates, reactants should disappear and product should appear as time goes by. As time goes by, the reaction occurs. Reaction means taking reactant A and turning it into product B. So as time goes by, A should go down and B should go up. In this case, we have a reaction that has two reactants. A and B are reactants, and P is our product. As the reaction goes through, as time goes by, the concentration of A and B should go down together and the concentration of P should rise. That is a chemical reaction that's occurring. Mathematically, and I'll never ask you to do this on an exam or a quiz, but it's important that we cover the concepts of it. Mathematically, what we're saying is when time changes, as time passes, the difference in time per unit time, the amount of A should go down, become negative, the amount of B should go down, become negative, and the amount of P should go up, or stay positive. So as time passes, A goes down, B goes down, and P goes up. That makes sense. We'll skip the actual rate equation. There's no real need, I think, to go into the true and deep math of this. It adds very little to your understanding, and in my mind, going over the rate equation and solving for it adds lots of confusion. This is the rate equation. The rate of a enzymatically catalyzed reaction is equal to a rate constant times the concentration of A raised to the power of F times the concentration of B raised to the power of G. If you really like that kind of stuff, it's explained in the textbook, but I will not ever hold you accountable for material around the mathematics of the rate equation in this course. This does bring us to something we do have to cover, though, something that is somewhat confusing, and that's the overall order of reactions. And before I go too deeply into this, I do want to say that there is a review video on the order of reactions already posted on YouTube. I put that together for last semester's class, and it seems to have helped many students. So if I go through this material and you find it to be a little bit much, you can kind of get a second pass, more informal description of rate orders and overall orders of enzymatic reactions there in that review video. So when we talk about an overall order of a reaction, what we're referring to is the number of molecules involved in the rate of the reaction. In other words, how many molecules actually have a say in how quickly the reaction occurs. How many different molecules influence the rate of the reaction? Or, said another way, the rate of the reaction depends on how many different molecules. We'll deal here in this course with first-order reactions, second-order reactions, and zero-order reactions. Do know that there are third, fourth, fifth-order reactions, and they're just insane. But we'll stick to these basic three here. So let's consider the simplest reaction we can imagine. A is converted into product. A is the reactant and P is the product. In this case, only A is involved in the rate. A becomes P. Add more A to the reaction, you'll get more P, and you'll get more P faster. Reduce the amount of A in the reaction, you'll get less P, and you'll get P less rapidly. So A is involved in the rate of the reaction. Only A influences the rate of the reaction, and so we refer to this as a first-order reaction overall. There's only one molecule influencing the rate. This is a first-order reaction with respect to A. In other words, from A's perspective, if there's more of it, the reaction goes quicker. If there's less of it, the reaction goes slower, and so it's a first-order reaction overall, and it's a first-order reaction as A kind of considers itself. An example of this, a real-world example of this, is the decay of any radioactive isotope. We can use P32 as an example. If you have more P32 around, then you get more decay products. If you get less P32 around, you get less radioactive decay. This is a first-order reaction. The only thing controlling how quickly and how much radioactive decay you get is how much P32 you have on hand. Let's move to our second example. Here we have two reactants, A and B, and they're converted into two product molecules, C and D. This is a second-order reaction overall. If you want more C and D, what do you need to do? If you want more C and D, you need to add more A and B. In other words, if you want more product, you have to add more of both reactants, therefore making this a second-order reaction. Both A and B are involved in the chemistry, both A and B influence the rate, and more of them will increase the rate of the reaction. Now, if we consider the rate of the reaction with respect to A, in other words, let's look at A only. If we keep B constant, and we only add more A, we're not going to speed up this reaction as much. The amount of B will become limiting. And so this is a first-order reaction with respect to A. In other words, from A's perspective, if only A is changed, then only one thing is influencing the rate of the reaction. The same is true from B's perspective. From B's perspective, if the amount of A is kept constant, then only influencing or changing B's concentration will influence the rate of the reaction. So with respect to B, this is also a first-order reaction. But overall, if we increase A and B together, we can get more C and D, making it a second-order reaction because two molecules can influence the rate of the reaction simultaneously. An example of a second-order reaction that we'll be covering much later in the course is the degradation of glycogen, our glucose stores in our muscle and liver. Glycogen is a tree of many, many glucoses. You can have thousands of glucoses on glycogen. If you add a single molecule of inorganic phosphate, you will release one of those glucoses and the glycogen will be reduced by a single glucose. So what this reaction is saying is you need two different reactants, glycogen and inorganic phosphate, and that will get you two different products, released glucose and glycogen minus that glucose. If you want more glucose released and smaller glycogen made, you should add more glycogen and more inorganic phosphate. Just raising one or just raising the other will get you a faster reaction, but not nearly as much faster as if you raise both of these reactants together. This is a second-order reaction overall because both the amounts of glycogen and the amounts of inorganic phosphate are involved in the chemistry and both contribute to the rate of the reaction. But just as in our more generic example, from the perspective of each reactant, keeping the other constant will make this a first-order reaction for each. So, while somewhat complex, those are also somewhat intuitive. What then is a zero-order reaction? Well, a zero-order reaction is when the rate of the reaction is completely independent of all of the reactant concentrations. In other words, raising or lowering the concentration of the reactants has no effect on the kinetics of the reaction at all. Enzymatic reactions can be zero-order when the concentration of the reactants, and again now we're going to start calling reactants substrate, because any time a reactant binds to an enzyme to be worked upon, that reactant is then called substrate. So when the concentration of substrate are so high that they saturate all of the working enzymes, that is when we have a zero-order reaction. In this case, the reaction is at full throttle, it's going as fast as it possibly can, and so you cannot make it go any faster nor any slower by manipulating substrate levels. Let's use here the example of cars going over a toll booth. We can think of the Mass Pike heading into Boston during rush hour. Cars are waiting in a long, long line at a 6-lane toll plaza to cross a 2-lane bridge. There's lots and lots of cars waiting to go through the toll plaza, and the toll plaza can only process so many cars per unit time. The toll plaza is the enzyme. Crossing the bridge is the product, and all these cars waiting in that line are the reactant or substrate molecules. Imagine a very, very long line of cars. Let's say the line of cars is 150 cars long. That's the concentration of substrate. If you add 10 or 20 cars to the end of that line, are you going to make the enzyme work any faster? Is the toll plaza going to process cars any more quickly? Of course not. Are cars going to cross the bridge any more quickly, becoming product? No, they will not. Let's go the other way. If you remove 20 cars from the end of that line, so you've gone from a line from 150 to 130, is that going to speed up anything at the toll plaza? No, it won't. Will cars go any more quickly or any more slowly across the bridge? Of course they won't. There's such a backlog of cars, there's such a backlog of substrate, that adding a little more or taking a little away has no influence on the rate of the reaction at all. Since the rate of the reaction is not influenced by altering the substrate concentration, this is a zero-order reaction. The rate of the reaction is constant, and adding or removing substrate is not going to have any effect on that rate at all. So to summarize this lecture chunk, The energy of activation is the amount of energy required to get the reaction to proceed forward. It is that mountain of free energy that the universe must input into the system in order to reach product. But now we're saying that the energy of activation is actually the amount of energy needed to reach transition state. It's the amount of energy needed to make this hybrid molecule that is no longer reactant on its way to being but not quite yet product. It is the bread dough of the reaction. All enzymes do is lower the energy of activation, and they do so by working on reactant, working on substrate, doing the work necessary to create transition state. That's what enzymes do to lower the energy of activation. They change nothing else about the chemical reaction. Reactant goes to product either way. We ended with some dry kinetics, some zero order, first order, and second order reactions, where we talked about how many molecules can and do influence the rate of a reaction as that reaction proceeds forward towards product. We'll end in the last chunk with enzyme substrate binding and tie a lot of these concepts together. But until then, as always, thanks for watching.